Welcome to This Explainer. Today, we're going to unpack one of the absolute most transformative scientific breakthroughs of our lifetime. We are diving deep into the fascinating biological origins and the frankly revolutionary modern impact of CRISPR.
Here is our roadmap for today. We'll start with what CRISPR actually is, look at nature's ancient immune system, trace the decades of scientific discovery, and finally explore the modern gene editing revolution and its classification system.
To set the stage for this whole discussion, I want you to imagine something. Imagine you had a pair of molecular scissors. Now, imagine these scissors could find any specific sentence hidden within a massive 3 billion-letter book and either cut it out, fix a typo, or replace it with something else entirely. Well, that is exactly what CRISPR-Cas9 does. But instead of a physical book, it's doing this to the DNA inside living cells. Okay, Section 1. What even is CRISPR? Let's decode this rather intimidating piece of scientific jargon. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeat. I know, that is a total mouthful, but this acronym actually refers to a very specific pattern of stored memory found right inside bacterial DNA. You see, it's a natural immune system that bacteria evolved over billions of years to fight off viruses. Which means CRISPR is not a human invention at all. It is literally an ancient biological defense mechanism. The truly extraordinary story here is that scientists realized they could repurpose this natural system using two main parts. First, you have the Cas proteins, most famously Cas9, which act as those cutting scissors we talked about. And second, you have a short RNA molecule called guide RNA. This acts perfectly as a molecular GPS, directing those tiny scissors exactly where to make the cut. Moving to Section 2, Nature's Ancient Immune System. To truly grasp how this works in a lab, we have to look at the microscopic warfare between bacteria and bacteriophages, which are just viruses that infect bacteria.
So this biological defense happens in 3 brilliant steps. First up, infection. This is when a bacterium somehow manages to survive a viral attack. Second, memory stored. The bacterium takes a tiny fragment of that invading virus's DNA and actually tucks it into its own genome, right between those characteristic CRISPR repeats. These stored fragments are called spacers. Third, immunity activated. If that exact same virus ever dares to return, the bacteria is ready for it.
Now you might be wondering, how does it recognize the returning virus? Well, the bacterium transcribes those stored memory spacers into short RNA molecules called cRNA. And these act perfectly like microscopic wanted posters. The wanted poster guides the Cas protein to constantly scan all incoming DNA, seek out the matching viral sequence, and literally cut it apart, totally neutralizing the infection. It's just amazing. This is actual bacterial immunological memory at work.
Section 3, Decades of Scientific Discovery. So, how on earth did we go from bacteria fighting off viruses in the wild to editing the human genome? It was actually the result of over 3 decades of pure, curiosity-driven science.
The timeline accelerates incredibly fast once the pieces start coming together. It began pretty quietly back in 1987 when Japanese scientist Yoshizumi Ishino serendipitously observed these unusual repeats in E. coli. Fast forward to 2007, and Philippe Horvath and Rodolphe Berengo, who were actually working at a yogurt company of all places, proved this was an adaptive immune system. Then the pace just explodes. In 2012, Jennifer Doudna and Emmanuel Charpentier published their landmark breakthrough, proving the system could be programmed as a gene editing tool. This huge discovery earned them the 2020 Nobel Prize in Chemistry. And just recently in 2023, it paved the way for the very first approved CRISPR medicine, Casgevy.
And it's so important to note that this wasn't the work of just one or two lone geniuses. Many researchers contributed absolutely critical pieces to this giant puzzle. For example, Ru Janssen, who coined the term CRISPR in 2002, or Feng Zhang and George Church, who simultaneously demonstrated in 2013 that CRISPR-Cas9 could be used to edit mammalian and human cells. Every single scientist involved added a crucial building block to get us here. Which brings us to Section 4, the gene editing revolution. Now, gene editing tools actually existed before CRISPR, so we really need to ask, why did CRISPR completely revolutionize the field?
The juxtaposition here is just stark. Older tools, like ZFNs and TALENs, relied on really complex protein DNA recognition. That meant scientists had to engineer entirely new proteins for every single genetic target. It was incredibly slow, taking months, and could cost upwards of $20,000.
CRISPR-Cas9, on the other hand, it relies on simple RNA-DNA base pairing. The Cas9 protein stays exactly the same, you just change the RNA guide. That costs around $10, and it is practically trivial to design.
Because it's cheap, trivial to design, and makes it incredibly easy to edit multiple genes all at once, a process called multiplexing. It truly, literally democratized gene editing. There's a commonly cited quote that sums it up perfectly. CRISPR has done for gene editing what the internet did for information. It made it universally accessible for biology labs worldwide. However, this rapid accessibility also led to a massive controversy. In 2018, Chinese researcher He Jiankui shocked the world when he announced the birth of twins whose embryos had been CRISPR-edited to disrupt the CCR5 gene, basically an attempt to confer HIV resistance. The global scientific community widely and swiftly condemned this experiment as premature and ethically unacceptable. It really highlighted the immense, heavy responsibility that comes with such an accessible and powerful tool.
Section 5. The CRISPR Classification System. As researchers surveyed more and more bacterial genomes, they discovered an enormous diversity in these systems. I mean, they are not all identical by a long shot.
They are primarily divided into two main categories based on their architecture. So Class 1 systems use complex, multi-protein effector complexes to make their cuts. They're super common in nature, but actually much harder to repurpose in a lab. Class 2 systems, however, use a single large effector protein, and this simplicity makes Class 2 incredibly ideal for biotechnology applications.
Within this incredibly useful Class 2 system, we have specific types serving totally different functions. There's the famous Cas9, which cleanly cuts double-stranded DNA. Then there's Cas12a, which creates staggered or sticky cuts and is widely used in DETCR diagnostic platforms. And there's even Cas13, which uniquely targets RNA instead of DNA, making it perfect for Sherlock diagnostics. Nature's toolkit is just astonishingly versatile.
And the mastery of this versatile natural tool belt paved the way for a monumental milestone in the year 2023. That year marked the regulatory approval of Casgevy for sickle cell disease and beta thalassemia by agencies like the UK's MHRA and the US FDA. This was it. This was the moment CRISPR officially stepped out of the laboratory and became a highly targeted, real-world human medicine.
We've seen how nature's ancient, microscopic defense mechanism against viruses was meticulously uncovered by decades of science, eventually becoming our modern medical miracle. But looking forward, it leaves us with a profound question to ponder. With CRISPR making gene editing as universally accessible as the internet did for information, how will we choose to navigate the future of human medicine and ultimately human evolution? Thank you so much for joining me for this explainer. I hope it sparked your curiosity.